Hello students, today we'll be talking about pharmacology of adrenaline. Welcome to BAN lectures once again. Let us discuss this pharmacology of adrenaline in detail. Now what is adrenaline? Adrenaline is basically a naturally occurring catecholamine. We may call it a sympathomimetic amine also. It is a biogenic amine or a true sympathomimetic drug. It is released basically from the medulla part of adrenal gland and it is a neurohormone. About 90% adrenaline is released from medulla part of adrenal gland. About 10% adrenaline is released from sympathetic nerve endings. Catecholamine means it is a benzene ring. You can see to this benzene ring two OH groups are attached at ortho positions. So this is catechol ring system. And here basically in the side chain you find NH2 group, amino group. One H has been replaced by methyl group. Methylation has taken place here. So it is a catecholamine. Now it was discovered in 1920th century. In 1895, Polish physiologist Napoleon identified it from adrenal gland. He considered it as an active principle. He called it epinephrine. It was isolated in 1900 by Jokinstree from adrenal gland, and finally it was being crystallized from adrenal extract by John Abel, and he used the term epinephrine is basically used as per United States Pharmacopoeia. As per British Pharmacopoeia, we use the term adrenaline. Now, adrenaline acts upon adrenergic receptors, sympathetic receptors. There are two types of adrenergic receptor, alpha and beta. It was Elquist who discovered adrenergic receptors. Alpha and beta are the two major classes of adrenergic receptors, and they have got subtypes also. For example, alpha receptors have subtype as alpha 1, alpha 2, and beta receptors beta 1, beta 2, beta 3 are the subtypes. They are present in different locations in the body. For example, in the blood vessels you have both alpha as well as beta receptors. Similarly, in the heart predominantly you have beta 1 receptors. In the bronchial tree we have predominantly beta 2 receptors, like that. So adrenaline acts upon both these alpha and beta receptors, produce a number of pharmacological actions, and some of them have a very good therapeutic application also. Now here we would like to talk about biosynthesis of this adrenaline. So biosynthesis does take place within the adrenergic neurons. This we have to remember. So after being biosynthesized within the neurons, they have to be stored within the neurons in vesicles. And finally, upon nerve stimulation, they get released from these synaptic vesicles, interact with different adrenergic receptors. So we can see in the next slide how biosynthesis actually takes place. It starts from phenylalanine. Phenylalanine is an aromatic amino acid. From the dietary sources we get phenylalanine. Phenylalanine undergoes hydroxylation, means OH group is introduced in the structure. Here OH group is added at the para position, and this forms tyrosine by enzyme phenylalanine hydroxylase. Tyrosine again gets hydroxylated and one more OH group is added to tyrosine. Now it is a catechol ring system, and this compound is dopa. Now we want to remove this CO2 group by a decarboxylase enzyme, and we get dopamine. Dopamine has a catechol ring CH2, CH2 and NH2. Now dopamine is being hydroxylated by dopamine beta hydroxylase. This is alpha position and this is beta position, and OH group is added at the beta carbon. We get norepinephrine. Then norepinephrine is methylated by phenylethanolamine and methyl transferase. Methyl group is transferred, one H is being removed and methyl group is added over here. We get epinephrine. So the first step would be here from tyrosine hydroxylation, then decarboxylation, again hydroxylation, finally methylation. We get adrenaline. Adrenaline is synthesized within the neuron. Here, this is the neuron. It is synthesized within the neuron. It is stored within the neuron in the synaptic vesicles, and from these synaptic vesicles it is being released upon nerve stimulation. Calcium ions play a very important role for release of these adrenaline or noradrenaline by the process of exocytosis. This is presynaptic nerve terminal and this is postsynaptic nerve terminal. These are the respective target agents, that is receptors. So adrenaline will interact with alpha or beta receptors depending upon present in the target tissue. Now, adrenaline interacts with adrenergic receptors as we have said just now. Now, these very adrenergic receptors belong to which class of receptors? They belong to G proteins, that is G protein coupled receptors. So, you have alpha 1 receptor, you have alpha 2 receptors over here. So accordingly you get different type of responses. There are different type of G proteins. For example, we will say GI proteins. Secondly, we say GQ proteins. And next we say GS proteins. So what happens is, if a substance interacts with GI proteins, that inhibits cyclic AMP pathway and it would produce inhibitory effects on the target tissue. Similarly, if the drug or the substance interacts with GQ proteins, that will stimulate phospholipase C pathway, that would increase the level of inositol IP3 pathway and diacylglycerol, that promotes muscle contraction, smooth muscle contraction, that promotes vasocontraction in large number of vascular buds. So we can see in the next slide, this is G coupled proteins, G. They have different subunits, alpha subunit, beta subunit and gamma subunits. Now you can see now GS, one of the type of G protein, if any drug interacts with GS protein, S means stimulatory, that would increase the level of cyclic AMP by stimulating the activity of adenyl cyclase. So this is stimulatory action, causes increase in cyclic AMP production. So just now we have discussed any drug interacts with GI type of inhibitory effect, and GQ again that will cause vasoconstriction by promoting this inositol phosphate pathway, enhancing the level of phospholipase C. So accordingly you have a different pharmacological action. So this is the beauty of G coupled protein. Now, actions of adrenaline depend upon the relative density and affinity of adrenaline for these receptor subtypes in a particular tissue. For example, let us see the cardiovascular effects. There is adrenaline leads to increased heart rate, enhances contractility, leads to vasoconstriction, vasodilatation in skeletal muscle mediated by different adrenergic subtypes. For example, beta 1 receptors, if they are stimulated, that will increase heart rate. It would enhance contractility. It would enhance vasoconstriction mediated by alpha 1 receptors. It would enhance sometimes skeletal muscle activity leading to vasodilatation mediated by beta 2 receptors. Similarly, respiratory effects are mediated by beta 2 receptors. They, if stimulated, they produce bronchodilation. Similarly, decreased release of inflammatory mediators and mast cells. That again, this is beta 2 effect. This would be required in bronchial asthma. For example, if beta 2 stimulation is being done in bronchial asthma, it will produce bronchodilation. Secondly, it would help in decreasing the release of inflammatory mediators. Similarly, some metabolic changes also, or metabolic effects, you can say glycogenolysis and gluconeogenesis in the liver is being stimulated if beta receptors are stimulated. Similarly, lipolysis takes place in adipose tissues, that is mediated by beta 3 receptors. Adipose tissues are rich in beta 3 receptors, and alpha 2 effect would be reduced insulin secretion if stimulated. Similarly, that would enhance the secretion of glucagon mediated by beta 2 receptors. The action of insulin and glucagon are physiologically opposite to each other. Next, beta 2 receptors, they are linked to GS type of G protein as we said earlier. So when they are interacted by adrenaline, that leads to stimulation of GS protein that stimulates smooth muscle of bronchi, vasculature and skeletal muscle as we said earlier, activates cyclic AMP, facilitates bronchodilation like that. Now, other effects would be dilatation of pupil. This is being mediated by alpha 1 receptors present in iris dilator muscles. So, adrenaline is a mydriatic. It leads to vasodilatation of pupil. The other effects would be causing sweating, piloerection, involving sympathetic receptors. These actions mobilize energy, elevate cardiac output, etc. during stress. Now, some of the pharmacokinetic adrenaline can be administered by intravenous route, intramuscular route, or subcutaneous, but not by oral route. So, upon intramuscular route injection in the mid outer thigh, the absorption is fast, reliable, and if given by IV intravenous route, there is immediate onset of action. If given by subcutaneous route, there is no doubt slower absorption due to local vasoconstriction. However, heat or massage may influence the rate of absorption. Then distribution of adrenaline takes place within the body, but it does not enter central nervous system in significant account. Low plasma protein binding, so far as metabolism is concerned, it is being metabolized by two important enzymes, COMT, catechol O methyltransferase, and monoamine oxidase, MAO. These are present in liver and other tissues. The various inactive metabolites produced are metanephrine and vanillyl mandelic acid. They are excreted in the urine. So it is excreted via urine, main route of elimination within 24 hours. Adrenaline has short half-life, 2 to 3 minutes after circulation. Now, clinical applications of adrenaline would be anaphylaxis. During anaphylaxis shock, which is a severe allergic reaction, adrenaline is the drug of choice. So, for certain life-threatening allergies, etc., adrenaline can be recommended. It relieves bronchospasm because it stimulates beta 2 receptors, produces vasodilatation, etc., that is required there. During cardiac arrest, since it stimulates beta 1 receptors that enhances contractility of heart, increases the contractility of, has a direct inotropic effect, increase the force of myocardial contraction that is required during cardiac arrest. And thirdly, local vasoconstriction. It is combined with local anesthetic in small amount lidocaine. It is for causing shrinking local blood vessels mediated by alpha receptors that reduces systemic absorption of local anesthetic and controls bleeding also. In other words, it increased the duration of action of local anesthetic, which is often used in dentistry. Adrenaline is combined with local anesthetic for increasing the duration of action of local anesthetic. Thank you.